I hope you're all doing well. And I'm feeling excited for today's session. I welcome you all, and I hope you are all doing well. It's my pleasure to welcome you all on behalf of MMN, Marine Mammal Research Conservation Network of India. MMN CNI is an informal scientific and collaborative network. It is dedicated towards marine mammal study, research, monitoring, and conservation of marine mammals in Indian waters. And the network connects researchers, conservationists, and public, encouraging towards sightings and strandings of marine mammals in India, um, and to support mainly the conservation efforts. We are especially delighted to have our speaker today, Dr. Robin Baird, joining us. And thank you all for being here and taking time to be part of this program. And about Dr. Robin Braid. He is a renowned marine biologist specialized in study of marine mammals, particularly whales and dolphins. He is an expert in studying the false killer whales in Hawaii region, and he is a research biologist with Cascadia Research Collective and has conducted extensive field research across Pacific Ocean. And, and his work focuses on understanding marine mammal behavior, spatial mapping and population structure, movements, and conservation of species such as big whales and false killer whales. He's especially known for his long term studies using photo identification and satellite tagging to track marine mammals in their natural habitats. He has extensively contributed to marine conservation efforts, helping to inform policies aimed at protecting vulnerable species and ocean ecosystems. With that, I'll hand it over to Dr. Robin, and we look forward to an engaging and insightful session. This is a recorded session, and the discussions and questions will be addressed at the end of the talk. I hope you all will enjoy the talk.
Great. Thank you very much. And before I get started, I just want to mention that the work that I'm going to talk about is really a large collaborative effort. There's been a lot of different individuals that have played a critical role in the field out on the boat, as well as a lot of people who play really important roles in the office, working with data sets and photo ID catalogs. This is a long-term program, so we've had funding over the years from a number of different sources, but primarily the NOAA fisheries in the US. And we have really important collaborators on a lot of the work with researchers at the Pacific Island Fisheries Science Center, Southwest Fisheries Science Center, several different universities, nonprofit groups, and others. Now, my interest in false killer whales has been around for a long time. I first had the opportunity to do a necropsy on a stranded false killer whale in British Columbia, Canada, back in 1987. And we had another stranding in 1989. And these strandings are really what got me interested in the species. There were first records for Canada, and so little was known about the species that even a single animal provided a lot of information on things like diet and persistent organic pollutants. It introduced me to Steve Leatherwood, who invited me down to San Diego in 1988, and we worked together to produce a report that was summarizing information on what was known about false killer whales worldwide at the time. Steve had a possible field project happening the next year in Hawaii, and he had hoped that he would be able to get funding for me to come out to join the project, but that never happened. So I had to wait another 10 years or so before getting out to Hawaii and starting to work. And in 1999, I was offered a position with the Pacific Whale Foundation on Maui as their research director. And one of the main reasons why I was interested in getting out there was the opportunity to work with false killer whales. And the work originally was started with small vessel work. We've used a lot of different boats over the years. These are just 4 examples of them. The one in the bottom right is the one that we more or less use for all of our work now. It's a 7.3 meter rigid hull Zodiac with a custom bow pulpit. I wouldn't recommend doing this kind of work in the boat that's up in the upper left, a little bit too small. Um, we when we're out on the water, we're not using systematic line transit survey methodology, and instead, right from the beginning, we've been trying to maximize the spatial extent of where we're able to cover in terms of depth and distance offshore, and doing so in conditions that are suitable for working with small odontocetes. So this particular map shows 5.5 days of survey effort, all based off the island of Lanai here in the middle. And when the weather was particularly good, we went out into deeper water to the west, and when the winds were stronger, we went into shallower waters to the east. And that is basically a reflection of our overall approach of trying to maximize the spatial extent of where we work while minimizing biases as much as possible. Now, this work is not just on false killer whales. In fact, we work with every species of whale and dolphin we see, and our approach has always been that depending on the encounter rate, we'll spend more or less time with different species, and particularly the species that we encounter least often are the ones that are our highest priorities. Um, and so false killer whales, while they've always been our one of our highest priorities, if not our highest priority for what we would find and work with, has never been a particularly commonly encountered species. And so we work productively on all species of marine mammals that we encounter, but focus on the rare ones when we get the opportunities to. Now, we have had a couple projects where we've had a combined land and boat-based effort, and that basically allows us to broaden our search effort and increase encounter rates. This is one example of a land-based team that was up on a cliff that was just over 200 meters above the water. And if you zoom in, you can actually see the truck and the people at the top of the cliff and our boat at the bottom of the cliff. The land-based team is effectively able to tell us whether or not there are any species of interest within their range, and allows us to work elsewhere when they're not seeing animals and expand our range with some confidence that we're not missing animals that are in their closer area. So this work did not start as a long-term project. I think it's important to say, and it started out with relatively limited technology. Initially, it was photo identification. We were using film cameras from 1999 through 2002, and didn't go digital until 2003. And the key right from the beginning, though, is that we've had a lot of citizen science contributions of photographs, and individuals who were working with tour operations or professional photographers or people that otherwise just spend time on the water that would take and contribute photos, and that really dramatically increased the sample size of photos available for our catalogs. Another fairly low-tech thing that we started doing right from the beginning was using suction cup attached tags to examine diving behavior. These were not D tags. These were, a simple time depth recorder in a syntactic foam housing. So they only cost a few $1,000s rather than $15,000 to $25,000 that something like a D tag does. And we collected information on diving behavior as early as 1999, our first year. We didn't do a lot of suction cup tagging because there's always the potential for the tags to come off and drift out far offshore where they might be difficult to recover. But we did get some information which was actually incorporated into a paper that came out earlier this year on diving behavior that incorporates this high resolution suction cup tag data with dive data that we later obtained from satellite tags. Now, we also started biopsy sampling pretty early on in 2000. And the purpose of the biopsy sampling was originally to collect samples for genetics, particularly to look at population structure. You can see where this arrow had just hit the animal and was bouncing back. And if you look closely in the photo, you can actually see there's a little bit of blubber sticking out from that arrow. In addition to the population structure, we get sex in the individuals. And then as the sample size grew, these samples were also used to look at relatedness among individuals. Recently, an epigenetic aging paper was just published, and it's also been used for cell cultures. Of course, these biopsy samples can be used for multiple purposes, and the skin for genetics, but the blubber could be used for persistent organic pollutants, and if there was extra skin, also for stable isotopes. And then in recent years, collaborating with Christy West and her graduate students at the University of Hawaii, the biopsy samples are also used to look at the body condition with blubber histology and hormone chemistry for reproductive and stress hormones. And this just shows a method of collecting biopsy sample with a crossbow off the boat with the animals when they're close. Now, in 2007, we also added satellite tagging to our repertoire. This shows a false killer whale approaching the boat. You can see the person in the bow crouching with an air rifle with the satellite tag in it. And false killer whale is going to come up on the other side of the boat here, and Daniel's going to deploy a tag here. Now, the advantage, of course, to the satellite tagging is that we're able to track the animals from longer periods of time and in areas where we otherwise wouldn't be able to go with the boat. Now, I will also point out that there's 3 separate photographers here, one up on the bow pulpit and one midships and one near the stern. And that's also been a really important part of what we're doing is having multiple photographers to maximize the chance of getting really good quality photographs from every encounter. With just a single photographer, always something could go wrong with their camera card, or maybe their settings were off. Whereas if you've got multiple photographers, especially if there's individuals on both sides of the boats, it just increases the chances of getting really good quality photos of every individual, which end up being extremely important. Now I'll show you an animation of the movements of some of these satellite tagged animals to give you an idea of how quickly false killer whales can move around the islands, and also how extensive their movements can be, both onto the leeward sides of the islands where we're able to work on the west coast, but also onto the east sides of the islands, the windward sides of the islands that are extremely difficult, if not impossible, to work off of in a small vessel. And even when working in large vessels, which No Fisheries has done, sea conditions are such that finding animals is extremely difficult, and working with animals is very difficult. Now, this animation also shows multiple individuals. And it turns out these individuals are from 2 different social clusters, which I'll talk more about in a few minutes. And the satellite tags, when there's multiple individuals tagged at the same time, can be used to monitor the interactions among individuals, and that can play a really important role in understanding everything from social organization to spatial use to determining what factors are driving use of space, whether or not they're social factors versus environmental factors. Now, this work, as I mentioned earlier, started in 1999, so we're, I think, in the 28th year now. Over the years, this map shows the survey of our track lines in yellow. We've worked off all the main Hawaiian islands for the last 24 years in a row. We've worked off of Hawaii Island. A lot of the work is concentrated off Hawaii Island simply because it has the deepest water close to shore and the largest lee. Working off some of the other islands, the islands are much smaller. The size of the mountains are much smaller, so they're much more subject to uh, inappropriate working conditions because of the strong trade winds that come from the east and blow to the west. But we have worked off Kauai 17 different years. A lot of the Kauai work has been funded by the U.S. Navy because there's a Navy training range off of Kauai. And off of Oahu, 8 different years, and Maui and Lanai, 12 different years. And typically, these field projects are us going out for anywhere from 10 days to 2 or 3 weeks at a time, going out on the water every day during that period. And then since 2020 with COVID, one of our crew who's based on Hawaii Island in Kona, when things shut down during COVID, he began doing what we call rapid response work. And when there were reports of false killer whales in particular, he would go out, or when the conditions were just extremely conducive to work, he would get out on the water. So that work has happened in between the more dedicated field projects. Now, we've encountered 18 different species of odontocetes in our work and 3 species of baleen whales. False killer whales are midway down that list. Over the years, just in 28 years, 153 encounters, although a large proportion of those encounters were a response of someone calling us about false killer whales, or when we've gotten a tag on a group, being able to relocate that tagged group later with the data coming in from the satellite tag. And so you can see if our goal was only to work with false killer whales, we would be spending a lot of money and accomplishing very little. But instead, simultaneously looking for false killer whales, we've done a lot with many other species as well. This shows the distribution of false killer whale sightings around the islands from multiple sources. It includes both our work and the offshore track lines are from Pacific Island Fisheries Science Center cruises. A lot of the track lines in blue around Maui are from Pacific Whale Foundation, we collaborate with. But you can see that from the sightings of false killer whales, they are distributed among all the islands. Now, false killer whales are social animals, and when we find them, they're typically in groups, although the groups can be spread out over extremely wide distances, 10, 20 kilometers. Sometimes you can find separate subgroups that are all coordinating their behavior and moving together. And when they come together, it's often for feeding. And in this particular case, this photo here, what you can see here in the back is one false killer whale, the tip of the dorsal fin of another false killer whale. And this much lighter gray animal is actually a broad-billed swordfish that's upside down. This is the tail fin from the swordfish and the anal fin. And this illustrates that they will sometimes feed on very large prey. This swordfish was probably about 10 feet long. They share their prey. This is part of another swordfish from the next day, breaking it up, passing it back and forth among individuals. And the fact that they're feeding on large prey and they share their prey is extremely valuable from the perspective of documenting what species of prey they're feeding on. It means they bring their prey up to the surface, and probably more so than any other species of whale or dolphin in Hawaiian waters, we're able to characterize the species of prey. This is a very large mahi-mahi that this false killer whale is carrying. In Hawaii, false killer whales are feeding on game fish of a variety of types, both the pelagic game fish like the mahi-mahi and the swordfish, but also reef-associated game fish such as this giant trevally. And over the years, we've documented through observations them feeding on 4 different species of tuna, swordfish, short-billed spearfish, moonfish, mahi-mahi, wahoo, and then a variety of others. And from strandings, the list here includes both observations and strandings. While strandings are relatively rare in Hawaii, they have provided both confirmation of some of the same species, but also documentation of a couple new species that hadn't been recorded through observations, and in particular, 2 species of squid. So it's possible that either the individuals that were feeding on squid were debilitated in some way and feeding on squid instead of pelagic game fish, or simply that these squid are being captured deeper in the water column and not brought up to the surface where we're able to document what they're feeding on. I mentioned that individual photo identification is basically the basis of everything that we're doing. It allows us to determine who's traveling with whom, which can be used to look at population structure as well as social structure, where individuals are moving and spending their time, as well as get information on life history of individuals, and in particular things like age of birth or calving intervals. And importantly, we're able to use these to estimate abundance through market capture abundance estimation. Now, one of the most important findings that emerged from the work was based on primarily on photo identification and then supported by the genetic work. This is a social network of false killer whales documented from 1999 to 2021. The animals highlighted in the red circle were ones that had all been seen associated together and photographed around the main Hawaiian Islands. And when we started satellite tagging these individuals, the map shows on the right that these individuals were spending all of their time somewhat in association with the main Hawaiian Islands. The furthest offshore we had them go was about 120 kilometers. And this group was recognized based on a combination of the genetics, photo ID, and tag data by NOAA Fisheries as the Hawaii insular stock in 2008. Now, there were also groups that had these isolated social clusters, ones that didn't link with the main component of the social network, and the satellite tagged individuals of those showed an extremely different pattern. You can see here the tracks moving offshore outside of the 200 nautical mile exclusive economic zone into international waters. And these were recognized that same year as the Hawaii pelagic stock of false killer whales. And just to expand out to give you an idea of how far these the Hawaii pelagic stock false killer whales are moving, one of the tag groups has moved over 2,000 kilometers away from where they were tagged into the eastern tropical Pacific. Now, there's also another component to the social network highlighted here in the bottom left. And these individuals, when they've been satellite tagged, have shown a very restricted range. Excuse me. But that restricted range has been in the northwestern Hawaiian Islands and overlapping with the main Hawaiian Islands just off of Kauai and Niihau, the western most of the main Hawaiian Islands. And these individuals were recognized as a second island-associated population called the Northwestern Hawaiian Island stock in 2012. Now, I've mentioned the genetic work. Mitochondrial DNA is one of the easiest analyses from the biopsy samples. It's passed on from a mother to her offspring, and the genetic work has been really important at confirming both the identity of these, the genetic identity of these different stocks, as well as for other types of analyses. So each one of these circles represents one mitochondrial haplotype, and the size of the circle shows how many individuals share that same haplotype, and the color is where they were sampled. So the main Hawaiian Islands population and the northwestern Hawaiian Islands population that I just showed you both share a common haplotype, which reflects that they share a common ancestor. And that means that these island-associated populations probably originated from the same single colonization event, and then separated into the northwestern Hawaiian Island and main Hawaiian Island populations. They do differ in their mitochondrial DNA ratios and in the nuclear DNA. And the pelagic individuals have very, very different haplotypes, a much greater diversity of haplotypes. And they differ in both mitochondrial and nuclear DNA from both the main Hawaiian Islands and the northwestern Hawaiian Islands populations. So genetic confirmation of what was becoming apparent based on the photo ID data and the movement data. Now, in terms of photo identification, for the main Hawaiian Island population, it's not just one mass of individuals that are all freely associating, and instead the social network analyses have revealed that there's 4 different social clusters. If these were killer whales along the west coast of North America, we would call these pods, but we call them clusters because our understanding of them emerged from these social network analyses that show them in these clusters. But these clusters basically represent long-term stable groups of individuals. When you restrict the analyses to those with a higher association index value, you can see that these clusters separate out pretty strongly, with 2 of them being very tightly associated groups and the other 2 having some linkages among them. And we've used the information on social cluster to get an idea or to examine the satellite tag data and get an idea of where they're spending their time. And we've discovered that the different social clusters have different high density areas. So this shows the high density area from satellite tag data for cluster 4. You can see this hotspot, high density to the southwest of the island of Lanai here. And this hotspot is one of the reasons why we semi-regularly have field projects that are based out of Lanai. It's the most easily accessible high density area for false killer whales around the main Hawaiian Islands. So you compare it to this other hotspot up here that's more or less on the windward side of the islands and in an area that'd be very difficult to work in. This is the density map for cluster 1 false killer whales. And by comparison, you can see the highest density areas are up in this channel here, which is extremely windy off the north sides of these islands, which again is extremely windy, and further offshore. And cluster 2, the highest density area is again in this very windy channel and on the windward sides of the islands. So as we were collecting all of this information that were giving us more on the biology of the animals and their population structure and their range, information started to become apparent that the population was undergoing a decline. And that evidence of decline came from a couple sources. Joe Mobley through the University of Hawaii had undertaken aerial surveys for humpback whales from the 1990s and 2000s. This map in the upper left shows the track lines for their aerial surveys. And while they were primarily focusing on humpback whales, they were documenting other species. And in particular, their sighting rates of false killer whales in the graph in the upper right declined dramatically between their earliest surveys and their later surveys. And by comparison, other species of small odontocetes, short-fin pilot whales, spinner dolphins, pantropical spotted dolphins, and bottlenose dolphins shown in this graph in the bottom left were not showing a dramatic decline. So this was evidence that the main Hawaiian Island population of false killer whales was declining. There's a variety of other evidence for decline. I had mentioned that project that Steve Leatherwood was planning in 1989 that he had hoped to get me to go on. Well, he and Randy Reeves had did some aerial surveys where they were particularly looking for false killer whales, and they found that the largest groups had over 400 individuals. It was the third most common species they were encountering, and that suggests that the abundance was relatively high back then. Dan McSweeney, who had been working off the islands since the mid 1980s, had noted a decline in sighting rates. There was also a decline in sighting rates off of Maui, and genetic evidence had indicated a recent decline in population size. So all of this evidence for decline basically led in 2009 the Natural Resources Defense Council to submit a petition to list this insular population of false killer whales as endangered under the U.S. Endangered Species Act. And in 2010, NOAA Fisheries released a very comprehensive status review of Hawaiian insular false killer whales, which showed that they were a distinct population, that population was significant in the global context, and that there was a variety of lines of evidence that the population was endangered and declining. And then in 2012, the population was listed as endangered. So how's the population been doing since it was listed as endangered? Unfortunately, not very well. This is a paper that came out last year that shows that since the population was listed as endangered, it's been declining at an average of about 3.5% per year. And the current estimate for the population is around 140 individuals for this main Hawaiian Islands insular population. Now, what could be causing the decline? And I think a lot of it comes back to what they're feeding on, which I've already mentioned is a combination of large pelagic game fish, as well as some reef-associated game fish. And one common factor for all of these is that they're species of fish that are subject to human harvesting. But there's other things. What they're feeding on are prey that are relatively high on the food web. We published a paper in 2020 that showed that all the adult males that have been sampled and about a third of the adult females had levels of PCBs in the blubber that exceeded levels that are likely to influence the immune system function or reproduction. And three-stranded animals that Christy West had examined had the highest levels of PCBs of all the individuals. So high levels of persistent organic pollutants could definitely be one factor influencing them. There's been a decline in the prey base over time. The top graph shows catch per unit effort for yellowfin tuna, and the bottom one shows the average weight for yellowfin tuna. The bottom time scale is an important one. Goes from the 1940s up until 2008, and the average yellowfin tuna is about half the size in recent years as it was in the 1940s. And importantly, in terms of the way fish are managed in Hawaii, there's no size limits for pelagic species. There's no catch limits in any of the nearshore fisheries. No observer programs or electronic monitoring in any of these nearshore fisheries. So there's just largely a free-for-all in terms of how fish are caught, and no particular effort to try to conserve fish populations. It's also possible that there's some level of deliberate shooting in retaliation for depredation. They're feeding on the same prey, and false killer whales have been known to take fish from fishermen since at least 1963. Many fishermen carry firearms, primarily to kill or deter sharks that are coming in to eat their catch or billfish. And there have been widespread reporting of fishermen shooting dolphins, not necessarily false killer whales, but other depredating species of dolphins. Marine debris is also an issue. This is a false killer whale from the endangered population that we documented last year with a line wrap around the body that's obviously cut through the dorsal fin. It's the first time we've documented marine debris associating or affecting false killer whales. They've been marine debris has been an issue with many other species of whales and dolphins, but this illustrates that they can affect false killer whales as well. Another potential threat to the population is Toxoplasma gondii, which causes toxoplasmosis. Cats are the definitive host for Toxoplasma, so if there's no cats in the environment, there's no toxoplasmosis. And of course, the Hawaiian archipelago originally had no cats, and they were all brought with the Europeans. The oocytes from Toxoplasma can persist in the ocean for up to 2 years, and they accumulate in filter feeders, which include myctophids, but also some other species of fish, including awa, which is milkfish, and that is a species that's likely prey of false killer whales. So there's a mechanism for transfer from terrestrial environments into the marine environment. And toxoplasmosis is actually known to be a significant threat to Hawaiian marine wildlife, particularly Hawaiian monk seals, but also spinner dolphins and bottlenose dolphins have been documented in Hawaii with toxoplasmosis as the cause of death. Now, the other very likely candidate for at least a proportion of the mortality and part of the decline is evidence of fisheries interactions. Now, I mentioned that false killer whales are feeding on the same thing that humans are trying to catch, and they will take fish off lines. We published a small paper in 2005 that showed that individuals in the main Hawaiian Island population had high levels of dorsal fin disfigurements, about 3.75% of the catalog. We looked at this again in 2014, 2015 with a much larger sample size, and the rate of individuals with injuries in the dorsal fin that were consistent with fisheries interactions was up to 9%. And then we looked at this again in 2024 and found that the rate of individuals with injuries that are consistent with fisheries interactions is up to almost 13% in the main Hawaiian Island population. We compared it with fairly large sample sizes to individuals from the northwestern Hawaiian Island population, which had just 1% of individuals with these types of injuries, which makes complete sense since there's virtually no fishing effort in the northwestern Hawaiian Islands. And the pelagic or open ocean population, which had just over 5% of individuals. And the pelagic population is known to interact with the offshore longline fishery. The other thing that's shown here that was new is that we looked at mouth line injuries, because these individuals are getting hooked in the mouth and then struggling against the gear, and so they're more likely to have mouth line injuries, and that's in fact what the results showed with about 17% of the individuals from the main Hawaiian Island population having evidence of mouth line injuries. And when you looked at individuals that we had both mouth line and dorsal fin images, the proportion of individuals in the population with one or the other of those injury types increased almost to 29%. So more than a quarter of the individuals in the population had evidence that they've survived fishery interactions. Now, these fishery interactions are widely distributed throughout the social network in the main Hawaiian Island population. The individuals that have the highlighted points here are those with evidence of fisheries interactions. So you can see it's not just one cluster or one subset of one cluster, but individuals from all the clusters. Cluster 3 in particular, 38% of the individuals have evidence of fisheries interactions. Compare that to cluster 2 with less than 20% of the individuals. So there seems to be some variation among social groups. Now, we've tried to use the results on evidence from fisheries interactions and information on where fishermen are fishing to identify the areas where a fisherman is most likely to have interactions. And these maps on the right show fishery overlap indices based on the number of days fished, and they're just represented in two different ways. In areas like Kona, where we work, are relatively low areas for an individual fisherman's likelihood of having an interaction, whereas these areas that typically reflect the hot spots for the false killer whales from the tag data are probably where an individual fisherman is most likely to be having depredation occurring and interactions occurring. And so we've argued that these areas would be the areas that would be most important to have either outreach efforts or monitoring efforts, observers or electronic monitoring to try to better understand the interactions that are occurring. And there's a lot of different types of fishing, hook and line fishing in Hawaii. And they are primarily targeting the same types of fish that false killer whales like to feed on, but in particular, trolling with lures, trolling with bait, various types of bait fisheries. The Ikashibi fishery is using squid for bait that they're catching at night. So a lot of different fisheries that could be interacting with false killer whales. Excuse me. But I included this photo of a newborn false killer whale just to illustrate that there is hope. The population is continuing to reproduce, and if the threats can be addressed, then that has the potential to rebound, and that's certainly what we're hoping for ourselves. So. I think I'm almost done with the slides and would be happy to answer questions. I think this one is just a nice video to ground us all at the end, show us one of the reasons why we work with these animals, and happy to answer any questions.
Thank you, Dr. Robin. It was a really great talk and very insightful articles and photographs, and I enjoyed the talk thoroughly. I would request the participants, if you have any questions, just put on the video and like you can interact with Dr. Robin. Anyone has any questions from the participants?
Yes, I have a question. Hello, can you hear me?
Hi,
Hi, I'm
Priyan.
Priyank Patel. Yeah. My question is, how will like shifting ocean temperatures and prey migration patterns alter the social structure and survival strategies of like different killer whale populations?
Yeah, so just to make sure I've got it correct, you're wondering about prey movements and oceanographic factors that might influence.
Yeah.
That correct?
Yeah.
It's a good question, and it's a difficult one to get at because there's often a lot more known about the movements of the false killer whales than about their prey. Even though their prey are commercially important, there's not a lot known. Well, there's a really diverse diet, and we don't know, even though we have a good idea of the species of fish that they're feeding on, we don't have a really good idea of the proportions of different species, and in part that's because some species are much easier for us to identify than others. Like mahi mahi, or you don't have to be a fish expert to determine that it's feeding on a mahi mahi, whereas there's 4 different species of tuna, and sometimes telling the 4 species apart is difficult based on the glimpses that we get. I think there's no doubt that there's going to be some influence of prey movements, and probably the best illustration for that is some recent work that was done by Michaela Cradafil, who looked at various oceanographic variables, particularly for pelagic false killer whales, to try to get an idea of what factors were influencing their spatial use, and she found that mixed layer depth influenced movement patterns. So if I've got it correct, when mixed layer depth was shallower, which is an indicator of productivity and potentially availability of fish prey, the movements of the false killer whales were more restricted. And also when chlorophyll A levels were higher, movements were more restricted. And when mixed layer depth was deeper and chlorophyll A levels were lower, the false killer whales were ranging more broadly, presumably because productivity was lower. So they're definitely responding to oceanographic factors, and that's sort of the best evidence that we have for it at the moment.
Thank you.
I have
Thank-
few questions.
Thank you,
if there is no-
Priyank.
Sorry.
Thank you,
can you hear
Priyank.
me? Sorry.
It's okay, ma'am. Please go ahead.
Yeah, so Robin, thanks so much for the talk. There were more than 60 registrations, but I'm not sure what's going on. I think the Shark International Conference and the IWC going on. But I had a few, I was very thrilled to see your survey lines. Because for some reason out here, the minute we say let's do a survey, we are either thinking parallel transact lines or zigzag survey lines. And these things are so expensive to begin with, that it really in a way, it really limits how much we can do. So I was curious about that, So I realized that y'all are lucky because the shelf is really narrow at some places and y'all can reach the deep waters really quickly. But was it always just random? Did y'all just leave and just go anywhere? Or did y'all have certain spots that you knew you wanted to cover? How do y'all decide where to go? Because the data is covering just about everywhere. The satellite tag was a great initiative in that sense because you could see them using all sides of the islands and everything. But when you did the boat surveys, when you started,
Yeah,
that was
well,
my-
yeah. When we first started the work, Jay Barlow was one of our early supporters, and I spoke with Jay back in 2000 about whether or not what sort of survey effort we should do. Should we try to do some sort of systematic effort? And Jay's point back then was that Hawaii is a fairly oligotrophic system with low density of animals. And these systematic line transect surveys, I think, are most valuable in areas where you have high productivity and high encounter rates. If you look at the abundance estimates that come from either photo ID or from line transect estimates, the variability associated with the abundance estimates in small populations is influenced by the resighting rates from photo ID, but from line transect estimates, it's influenced by the total encounter rates. And so in a low density area where low encounter rates, you're going to have very high variance associated with estimates. So part of it was that those low encounter rates and the photo ID was going to be a better way to do it. That said, you can't photo ID animals in the Beaufort 4 conditions, whereas you can in Beaufort 1 or Beaufort 2 conditions because of encounter rates and waves and splashes and so on. So we've always tried to spend our time in areas where the conditions are best, but and sometimes that's near shore. but we take advantage of the rare opportunities when it is calm to get offshore, to get to areas we wouldn't otherwise be able to go. If weather was not a condition or a consideration, and we're going out of one particular harbor, we would go north one day, south the next day, offshore the third day, but because weather is sometimes uh, a consideration, and we're forced to go south more often than not, um, we will still try to mix up our survey effort to minimize biases. So what I mean, or one example of that is if we're forced to go south because of the strong trade winds, we'll go near shore in the morning and then offshore in the afternoon on day one. And then the second day, we'll go offshore in the morning and near shore in the afternoon. We keep all of our survey track lines during a particular field project on our GPS, and we try to minimize overlap track lines. So all of it comes down to trying to minimize biases. You asked about the satellite tag data. We do know from the satellite tag data now where the animals are more likely to be found than others, but it's still very difficult for us to get to those areas because most of the high density areas are um, in windy areas. So we're forced to um, work in other areas. But again, when the weather is good, then that's when we're expanding the reaches of our survey effort.
Yeah, that's great. Thanks so much for that. Because just, it just shows that even if we don't do the so-called sophisticated set of lines for small, even if it's a low density population, we are often told that we should be doing only that. So I think it's a great example to show that such good work can come out of it. I think Afa has a question. I'll let her ask because I have quite a few other questions actually. So I can go later.
Hi, you're right. Thank you so much for the talk. I'm Aoife. I'm a research master's student based in St Andrews, and I am studying false killer whale acoustics. So I really wanted to come and hear everything you had to say. I was just wondering, in your many years of experience with false killer whales, specifically out in Hawaii, whether you have noticed any major differences in acoustic production rates or the types of sounds that you're hearing, depending on the population or in those hotspot locations compared to other locations, anything like that.
Yeah, well, the acoustic work that I've been involved in has been somewhat limited, and it's mainly been back in the early days, we were getting acoustic recordings before they had been really well characterized in order to allow them to be well characterized. And that was work that was done by Simone Bauman-Pickering. We've also done a little bit of acoustic tagging work and provided those to Brie Madrigal for her recent paper. But they're extremely vocal all the time, I would say. And I'll pass on something that Paul Noctegal mentioned, which is Paul, who's worked with false killer whales and other species in captivity, has mentioned that their sounds are extremely loud and will travel a very long distance, and that I think is really important for some of the social interactions and the really widespread groups. I think that they're probably able to coordinate their behavior over very large distances, and particularly in an oligotrophic environment, the groups have to be really spread out in order to be able to find and locate dispersed groups of fish. Yeah.
That's great. Thank you so much.
Thank you, Ifa. There is one question by Tanya. Do you think tourism on the main Hawaiian Islands could also contribute greatly to decrease in false killer whale populations?
Yeah, and I saw a question in the comments asking about behavior with boats as well, and I think that's somewhat related. If you look at the high density areas where, well, there's no doubt that boats can sometimes cause problems, and the most obvious example is that at least some species of fish will hide underneath boats when they're being attacked by false killer whales, and particularly mahi-mahi. And so if a boat could interfere with individual predation events, false killer whales are chasing the mahi-mahi, and the mahi-mahi gets up underneath the boat, then that may effectively take that fish away from the false killer whales that were trying to feed on it. I think that that's relatively rare. And when it happens to us, we actually just move away from the fish to allow the whales to get it. False killer whales in general tend to be either indifferent to boats or interested in boats, and they'll often come over and bow ride, and in those cases where fish will hide underneath boats, false killer whales will come right up underneath the boat to get it. They don't seem to be disturbed by the boats in that sense. Some species are obviously much more sensitive to either boat noise or boat presence, and won't get really close to them. The other reason why I think tourism in general is probably not a big issue is because the animals are spending so little time in areas where there are actually a lot of tourists. And again, it's the tour operations are in Hawaii are primarily off one particular coast of Oahu, one particular coast of Maui, and one particular coast of Hawaii Island. And all three of those areas are relatively low density areas for false killer whales. And I'll give you an example. We had last October, we satellite tagged a couple false killer whales from cluster 4. And I don't know if you remember the map for cluster 4, but it was the one that had the fairly restricted range. And we had one of the tags that lasted up until last week, 192 days. And in that 192 days, there was only one day where that group of whales went off of the west side of Oahu where tour vessels could find it. And to the best of my knowledge, I think that one day may have been the only day in the 192 days that any tour vessels interacted with that group that we're aware of. And when I say we're aware of, we work pretty closely with as many tour operators as we can to try to encourage them to take photos and contribute those photos. And it was just that one day out of a 192-day tag deployment that we received any photos from that group. So I think the proportion of time that any particular group actually spends around tour boats is probably extremely small. That example is a bit of an extreme, but just a fresh one because the tag just stopped transmitting last week. But in general, I think unlike some other species where tourism is definitely an issue, I don't think it's a big problem with false killer whales in Hawaii.
Thank you, sir. Shripad, do you have a question?
Yeah. Hi, Dr. Baird. Thank you for the amazing talk. Your satellite tagging videos were really interesting to look at. It made me think about, how different were the sorts of preys that the different three populations go after. So like the Northwest population, the main population, and the offshore population. Because I also noticed in one of the videos that some of them are frequently going near the shelf region of the islands. So I was wondering, is there any difference in that, and how different are the preys?
Yeah, it's a good question, and we know, well, broadly, we don't know the answer to that question because we have relatively limited sample sizes of predation events by the northwestern Hawaiian Island population and the pelagic population. That said, when we have been around those other two populations, they're still feeding on large pelagic game fish, but that doesn't mean that they're not feeding on different mixes of them. In the 2020 paper on stable isotopes and persistent organic pollutants, we do have in there some evidence that different clusters within the main Hawaiian Island population have slightly different diets. And Michaela Cradafil, who I referred to earlier, just finished her PhD in March. One of the chapters of her PhD for the main Hawaiian Island population is currently submitted for publication right now, shows that there's broad overlap within that population among the four clusters, but one of the clusters in particular has a wider diet breadth than the other three. So similarities, but some differences as well.
Thank
And
you, sir.
I should add, let me just add to that question. Probably the best way I think of getting at diet composition is probably something like fecal samples. So either from stranded animals, Christy West, who does a lot of the stranding response, what they do when there's a dead stranded false killer whale is they will take fecal samples from multiple locations throughout the digestive tract, and then you can use genetic methodology to determine what species of prey were found in different parts of the digestive tract. And I think that will probably be, when there are strandings, that will probably be one of the most representative ways to see what they're feeding on compared to sort of our occasional observations of diet.
Okay, I just thought that because you had you had a pretty good list of, the different species that they go after. But I think that, again, like you mentioned, that was probably more for your main population.
Yeah, the majority of our encounters are with the main Hawaiian Island population. So the majority of the prey are from the main Hawaiian Island population.
Because they do seem to go behind a really big range of, quite generalized.
Yeah. Yes. Yeah.
Thank you.
So there is a question like, for how long does a satellite tag remain attached to a whale? By Tanya.
Yeah. So the tags on average, so these are limpet tags. This is an example of one of the tags. They attach with 2 surgical grade titanium darts. The average attachment duration for false killer whales is about 45 days, but there's a tremendous amount of variability. So I mentioned we had, we had one tag that just transmitted 192 days. I think that's our 4th longest or 3rd longest ever for a false killer whale. The longest for a false killer whale we've had, I think, was around 230 days. But because there's such variability, it also means that we get some that are 10 days or 2 weeks. We just had excuse me, the tags that we use, we have 2 different types, typically a location-only satellite tag and one that also records dive data. And the dive data tags, the batteries don't last as long as the location-only tags, but we just had one that records dive data last 60 days, which we were pretty happy with. That's the longest we've ever had a dive data tag last.
Thank you, sir. There is one more question. Like, are encounters between killer whales and boats including aggression or play, a sign of behavioral adaptation, stress response, or curiosity driven learning.
And is this question is for false killer whales, not killer whales. I think that the other thing about false killer whales in boats is that um, they they may associate boats with food because they take fish off of lines. Um, so I think that they could be approaching boats for that reason as well. Although in our case, I would say, well, false killer whales with people in the water are often show very interest in people in the water and approach people in the water, um, and their interactions with boats. I certainly have never heard of with false killer whales any reports that could be considered an aggressive interaction between false killer whales and a boat or false killer whales and people. There, the interactions I think are curiosity or play for things like bow riding rather than any sort of aggressive interactions.
Thank you, sir. Um, yes, ma'am, Dipani, ma'am.
Yeah, I had a list of questions actually. But we can go through them quickly. I know it's getting late. I just wanted to know if there were any indigenous communities there, historical fishing methods that may have changed, any positive, interactions between fishing communities and cetaceans historically.
Yeah, certainly. Well, there was no written history prior to the Europeans, and I think there was a lot of attempts to try to limit the Hawaiian language and so on. So I think there was a lot that was lost. That said, the going back, there's at least one case that I'm aware of that was reported in one of the very early U.S. exploring expeditions that involved a drive of a group of melon-headed whales by native Hawaiians back in 18 something. I don't know of any names associated with false killer whales or some of the other species. There's Hawaiian language names for spinner dolphins or perhaps dolphins in general, and for sperm whales and for large whales. But if there were names and relationships for some of the other species like false killer whales and pilot whales, those have been lost to history. A lot of the fishing methods that are there's certainly many indigenous fishing methods associated with reef-associated fish, and including a lot of fish ponds for fish farming effectively in nearshore areas. But no, like a lot of the history, there's no record of interactions going back. So it's hard to say whether or not that means that those relationships, those interactions didn't occur, or just that they've been lost to history.
Yeah. And I'll just ask one more question. I won't cover all those. It's just the signature whistles. If they're so resident and they're so tight knit, do we know if they have signature whistles? I mean,
yeah, I'll refer you to a paper that was just published earlier this year by Bree Madrigal. No evidence of signature whistles at this stage, but that doesn't mean they don't have them.
okay. Yeah. All right.
Yeah.
And the toxoplasma stuff was really shocking. I had no idea that it could get transferred. I mean, out here, we don't even have simplest things in the assays. So is it just something that the vets do? Is that normal for them to do, or was it?
Yeah,
part of the, yeah.
There's two things that have been done recently. One is an assay test to look for evidence of immune system response to toxoplasma exposure, and that work I can send you. There's a paper that came out in the last couple of years. I can send you that paper, but that work has shown that a number of species, including what you would definitely consider open ocean species in Hawaii, have had evidence of exposure to toxoplasma. And then the second thing that's been done is cases where an animal has from the necropsy has what's acute toxoplasmosis, and so toxoplasmosis was the cause of death, and there's lesions associated with that through many different organs, and basically from the histopathology work that's being done, they've actually been able to conclusively prove, well, maybe it's not histopathology, from the tests that conclusively document toxoplasma in many of the different organs. So it's sometimes it's just exposure, and other times it's the cause of death. In this paper, it mentions that I think 3 different spinner dolphins now have been known to have died due to toxoplasmosis, disseminated toxoplasmosis, and Christy West has extrapolated that out to more than 60 individuals in the population that would have died from it based on the carcass recovery rates. So in the case of spinner dolphins, it's probably a pretty significant cause of mortality. Bottlenose dolphin has also died from from disseminated toxoplasmosis, and that may extrapolate out to 15 or more bottlenose dolphin deaths based on carcass recovery rates, and then based on detection of it's been documented in a number of other species as well.
It's really shocking, and we really need to ask our vets here to do that one, because they certainly do it, given that cats are great pet. Most of the vets are trained for dogs and cats and things like that.
Yeah.
Yeah. All right. I won't ask the other questions. It's fine. I think there is a question in the Zainul. Do you want to ask yourself? Would you like to put on your video and ask Robin your question?
Yeah, that's fine. Hello. Am I audible?
Yeah. Yes.
Yes.
Yeah. Thank you for the section. It's really good. I really enjoyed the topics, and I strongly believe that by non-invasive photogrammetry work for in marine mammals for India is very much important. Like we have certain limitations to do the research in marine mammals. So, sir, I have a few questions. Like in your work, did you ever use the species distribution modeling to find out the hotspot? And another question, like in your second slide, I just noticed there is a white circle marking on the dorsal side of the animal. Is that because of any cookie cutter, like blisters or something else due to interspecies interaction or something else, or because of aging? I'm just curious about that to know.
Yeah. In terms of the wound, yes, they do certainly get bitten by cookie cutter sharks, and for false killer whales, when they heal, those repigment back to the background coloration. But that one image was a fresh cookie cutter wound. In terms of the species distribution modeling, that's something that's been done quite a bit with false killer whales based on the line transect survey results. So this is an effort led primarily by Pacific Island Fisheries Science Center, and they have produced species distribution models to estimate density of the pelagic population, and have found what factors are influencing they use a variety of environmental factors to try to see which ones are important in terms of influencing their spatial use. Probably the closest that we've done is again, her PhD is available on the Cascadia Hawaii publication page. If you go to it, Michaela Cradifield's PhD, one of her chapters is particularly on pelagic false killer whales based on our satellite tag data. And she has done a variety of environmental modeling of the satellite tag data to look at which areas they're choosing compared to what are available. And so it's different but similar in terms of identifying what factors are influencing their spatial use. And I would encourage you to check that out. You also had mentioned in your comment about aerial photography. We do some work with drones, and in particular trying to get aerial photogrammetry images. And there's a PhD student at University of Hawaii, Jens Curry, who has been doing a lot of drone photogrammetry of false killer whales, in part to look at how body condition changes over time. And that work, I think, is quite important, but a lot of these come back, whether or not it's the genetic work, the aerial photogrammetry, or the satellite tagging, what ties them all together is the photo identification work and knowing those individuals and being able to interpret all of those other results as they relate back to the individuals and the social structure. So the least expensive part of the methodology, taking a camera out and getting good ID photos, is the basis for interpreting all of the subsequent studies, and including the acoustics. I should add the acoustic work that Brie Madrigal just published earlier this year, again, knowing who those individuals are, knowing what social cluster they belong to, knowing that you've got recordings from different individuals rather than the same individual, and so on. So,
Yeah, that's like a dream. I don't know when. Just to have all these layers, along with the photo ID and the sex of the animal is just outstanding, I think. And I hope that the future generations in India will catch up and be able to do that. We are all still very being by funders being driven to population estimates, but slowly.
Yeah. Well, and I will add, the false killer whale work my first funding in Hawaii was to do some work with humpbacks. My second funding in Hawaii was to do some work with bottlenose dolphins. My third funding in Hawaii was to do work with beaked whales. The point being that we didn't get any funding to work with false killer whales until much, much later, but we were doing work with false killer whales the whole time. And I think that that's important, that while the funding may be for one very narrow question, that shouldn't limit what other types of associated information can be obtained at the same time. And the way we got funding, I mentioned that order from humpback whales to bottlenose dolphins to beaked whales. The way we got funding to work with beaked whales was by working with beaked whales before we had any funding to do it, and the same goes with the bottlenose dolphin work. While we were doing the humpback study, we were also getting bottlenose dolphin data, and we used that initial bottlenose dolphin data to then get money to do work with bottlenose dolphins. And while we were doing that, we were also working with false killer whales, and we did our first tagging of beaked whales prior to having any money to tag beaked whales. And these were relatively inexpensive, reusable tags that allowed us to then get funding for it. So that's been a part of the history all along of starting the work prior to having any specific funding for it, and that then leads to funding later. So
that's lovely. Thank you so much. Oh, Zainul has another question for you. Sorry, they're not letting you
sure.
go.
Doctor,
No worries.
thank you for the answers. And Doctor, I was just confused about a few things, like in the sense of the pygmy killer whale and false killer whale, and these are morphologically similar characters. While you are in the survey period, like in the field, so what are the key techniques we will use for like their social behavior, and also like in pygmy killer whale, they are nocturnal feeders, so they will not be that much active in the daytime. So in that kind of any characters of our animal, like this one, you have any that kind of strategies or trick while you are planning the time or the spot of survey, the area or the time of the survey?
Yeah, the those types of species specific behaviors haven't really influenced the area or time of the survey. I mean, going back to the difference between what we were doing and what other people were doing, which I didn't really mention. When I started working in Hawaii, there were lots of people working on humpback whales, and there were lots of people working on spinner dolphins. And what we did differently is not spend all of our time in areas where we were going to see humpbacks or spinner dolphins, but try to get into deeper water. So that's number one, getting out into deeper water when the conditions were good. And even when we got out into deeper water, the species we encountered most often were short-fin pilot whales, but we didn't focus just on short-fin pilot whales. We focused on whatever we encountered, and if we found pygmy killer whales, we spent as much time with them as possible, or false killer whales, we spend as much time with them as possible. So those initial decisions, not just studying what everyone else is studying, getting out into deep water when the conditions allowed, not just studying the most common species, but focus on the rare species when you see them, since there's not much of a lost opportunity cost because they're rare. The biggest thing, I think, for the differences among species, the thing that took the longest was learning how to productively work with some species that are very sensitive to boats. And pygmy killer whales are a good example, as are dwarf sperm whales. And having the patience to work with them without disturbing them. And once you disturb the animals, you're accomplishing nothing good. One, you're disturbing them, but two, you're not going to be able to get close to them again once they've been disturbed. So a lot of it is learning how to work with them without disturbing them. And in the case of pygmy killer whales, are an unusual one because if you approach them in a boat the way you would approach many other species, they're just constantly avoiding you. They're not avoiding you at high speed. They're just staying away from you. But I discovered after a while that if you position the boat, you spot them, identify them from a distance that you think they're pygmy killer whales. And then if they're pointing in one direction, often we'd go up in front of them and turn off the engine. And that would often result in them getting curious as to why this boat has turned off the engine, and they'll come right by the stern. We biopsied pygmy killer whales at least once with the engine off as they came over to the boat. And the same with dwarf sperm whales. They're very sensitive to changes in engine speed, or and beaked whales as well, putting the boat into gear and then back into neutral. They're very sensitive to those changes in sound. So learning that took a long time. And once we learned that, then, our ability to get close to them increased dramatically. So, lots of species differences in terms of how they're approached, but not necessarily where we look for them, as long as we're looking out in deep water when we get the opportunity to get out into deep water. So.
Thank you, Doctor. Thank you. And Doctor, one more thing. I just want to know, is there any studies based on the biological trait analysis of marine mammals, like in invertebrates like macrobenthos organisms? We are doing morphological trait analysis and biological trait analysis. So they find their functional complexity, how functionally rich the ecosystem. So if we have a country like India, so in our EZ, the marine mammal functional richness to make a generated the results like with a functional composition and also the redundancy like things with their morphological features or lifespan or other functional factors like there is some database like polytrites or something. They for the macrobenthos they done almost 41 traits list. So for similar that way in marine mammals, is that possible to we can build such a trait models, their fin portion, like their mouth portion, like something else, and their lifespan, and the area where they being, and pelagic or the depth where they're being, like that we can do like a develop a the data and through the fuzzy coding and things. Is that possible in marine mammal research?
Yeah, it's a good question. I don't know the answer. In a lot of the species that I'm working with, they're still at a very early stage in terms of understanding a lot of the basics of them. Not all of them, but some of them are just so rarely studied around the world. Pygmy killer whales and dwarf sperm whales are two great examples of that. Trying to more broadly characterize the role of all of these different species, I think is just, we're still at a pretty early level for that. So, but anyways, well, thank you very much. Thanks for everyone, and enjoyed the discussion.
Thank you so much.
Thank you so much.
Thank you. Thank you, Dr. Robin, for an insightful and engaging session and sharing all your experiences like slowly, and you answered us taking a time and effort into explaining everything so clearly. And we all had a very valuable experience of Hawaiian false killer whales. And it is grateful for the opportunity to hear from you. Thank you, Dr. Robin. And
Yes,
I
my
thank all
pleasure.
the participants for participating in the series of talks. Thank you. Thank you all.
Thanks, Robin. Have a good day
Right.
there.
Thank you. Take care. All right.
Bye.
Bye-bye.
Bye.